This is a beautiful building. I noticed while I was using the facilities that the math department is here and they have a storage room, and I'm assuming that's where they keep their random numbers. I'm particularly honored and pleased to be selected for this award. Prior to this, I had assumed that my major outstanding contribution to Harvey Mudd as a student was my role in raising the reputation of the school for its in the areas of social and sartorial awkwardness. Now, I should admit, at this point, 2015, Harvey Mudd has a significant reputation worldwide. At the time that I entered, 1971, it was really not known at all on the East Coast. I'm from Boston. First day of class, Professor Seven is going around the room pointing out, you're from here, you're from Hawaii, looks at my blue button-down shirt, you're from Boston. I later learned, or it occurred to me, of course, and a trick that I continue to use is these name tags are a big giveaway. But actually, I really did not intend to apply to this school. I thought I was going to Harvard Mid. Just checked the wrong box. So reading does matter. I was recently, I ride my bike to work most days, and a number of years ago I was riding along, hey, how you doing? This guy goes, outstanding, never been better. Since then, the world's divided into those who ask me how I am and those who absolutely refuse. So we talked about sacrifices in order to get this award. This is my wife, Julie. I will be, and she's right up here. In terms of actual sacrifices, I think more credit goes to her. Stavros Duesenberg, who I'll be speaking more of in a few moments, told me, never marry anyone exactly like yourself. One of you will be redundant. Julie is smarter, much more analytical and logical than I am, and considerably more organized. There's no redundancy in those areas. She's currently a piano teacher and neighborhood activist. These are my two children, both Elizabeth, who will be graduating next week, and my son Adam. So we will have two launched children with jobs. Now, in terms of people who really made a difference in my life, Bob Borelli and Stavros Duesenberg. Despite the ability to find anything on the web, I could not find a picture of Stavros. So I'm sure there is one, and it occurred to me later I probably would have found one in the yearbook, but I'd already sent this off. On Thursday night, John Russell and I opened the very last bottle of wine that Stavros and I fermented. 1976 Petite Syrah. It was still more or less drinkable, but it was decidedly enjoyable. We also opened a bottle, appropriate to our class year, of a 1975 wine acquired from Bob Borelli. Bob, as you may remember, for those from that time, he had his office on the first floor of Kingston Hall. He was frequently there at night, and I would frequently go and knock on his window. He would let me in, and we would talk, drink wine, and so forth. Another person not up here who I should credit, and I can't unfortunately pronounce his last name, but Sadat in engineering. Exactly. After that, although I came here as an engineer, that convinced me that I shouldn't be one. And Bob talked me into a major in chemistry, and that I would do as my senior thesis something on the chemistry of wine. And that gave me at least a little focus in my life of what I was going to do. I ended up doing something with NADH reductase, I don't really remember. But it was something else with Paul Van Eyck. And I did learn there that I liked perhaps toys or equipment more than I liked some of the chemistry and actual other kinds of things. And I kind of saw that I did have a bent towards instrumentation and analysis. But really, without Bob, I doubt I would have made it through Harvey Mudd. And he remained, although we didn't really remain close in person, we certainly remained close in spirit. And I should say, until long after I graduated, I always thought that Stavros' wife's name was Bunny. My career through the years started at Harvey Mudd. I took one of those extra year off. Since Maria opened this door, I will say it, when I first began thinking of what the contributions to my life's work and career were from Harvey Mudd, the first thing that came to mind was, at Harvey Mudd, I really burnished my relations with both anxiety and insecurity, and really it brought it to a fine point. And I only wish that the availability of the mental health ability to recognize that in fact there was help and possibilities for this kind of thing. It really made a better living, a better life. Now, relating to that, as a chemistry major, I also recognize that there is better living through chemistry. So from Harvey Mudd, I went on to graduate school at Johns Hopkins, the Uniformed Services University, the military medical school, and then on to biotech companies, and then finally onto my own, into my own company. I recognized in this transition period around between 1993 and 1998 that I really was not an academic. This was really not what I wanted to do. I just could not, first of all, I just could not remember enough to be a good academic and the type of details and types of focus. And secondly, I just didn't care. But what happened in '93 is I began to shift from more of the academic stuff, where I was working with academics, to very applied things. I was still making things. My father was an engineer, part of why I ended up, Harvey Mudd was on the approved list of schools that he would pay for. And so I still have that. I call that my engineering gene, since I like to make things. And that's what I do, but I'm working at a molecular level. In terms of another part of the career arc was a course that I took from Dan Peterson, and I believe it was biophysics. I really don't remember. It was kind of an oddball course at the time. And I learned about cell membranes, which cover mammalian cells, bacterial cells, pretty much all sorts of life. And this was really, the models that were coming out for this were very interesting and caught my attention. And that caused me to apply to the Johns Hopkins Biophysics Department. I selected that over many other prestigious other universities, in part because after being at Harvey Mudd in a small classroom environment, I was kind of terrified about going to a big school. Once I got to Johns Hopkins, following the burnishing to those two elements that I mentioned before, kind of really went off track as I lost the focus of not having those firm goals of pass this course and move on to the next thing. And I moved into another career path. People say that graduate school and postdoc gives you many opportunities for freedom and exploration. I would say one of the characteristics of my career is in terms of other people, having mentors, people like Bob Borelli and Stavros, who took me under their wing, and even though I didn't, to my mind, seem to have much promise or direction or anything else, still were willing to guide me along and help me along. Other mentors worked in a different way, and they pretty much found that I was more trouble than worth than it was worth to try to direct me in any way and just left me alone. And that was true for my graduate career. And although there was discussion about throwing me out because I didn't really seem to have much academic pressure, a professor who really didn't like me that much, but liked to be stubborn and difficult as well, insisted that they took me in and it shouldn't matter what I wanted to do when I got out, and I'm thankful to him for keeping me in. One of the things in, since Scott's here, we can say that was Warner Love. One of the things, as I was just so insecure and shy, it was just so hard for me to get up, talk about things, questions of. As a performer, I had no problem getting up in front of people, large crowds, and doing whatever. On top of that, the type of performing that I mainly did was what's known as table magic, where you walk up to complete strangers in a restaurant who aren't expecting anything, just there for a meal, instantly get them to like you and give you money. So it's really one of the hardest things. It's not like a piece of entertainment where you go to a show, you're buying a ticket, and they want it. So it's really this kind of weird difference between these two areas where one, I just couldn't do anything and was so insecure in this other one. Gradually over my career, I've been able to melt these two things, and it's come from confidence and developing some actual skills. And this area of being able to translate my enthusiasm for performing and entertaining into my work life. At the Uniformed Services University, and this is where a little magic comes in, one of the two people who interviewed me, I always did a little trick as part of the interview process, and I wouldn't tell the guy how it was done, so he had to hire me. This turned into a very productive period of my life, where I mostly drifted, but it was nevertheless very productive. And I mentioned before about membranes. So membranes have a lot of stuff stuck into them. They're antibodies that will link against them. And there's some antibodies that, on a B cell, the cells that make antibodies, will, when you bind to them, it sort of stimulates them, gets them doing things. And the group that I worked with found that if you make polymers of these, that is, you put many of these onto a long string, it had a completely different set of properties in activating a cell. And so I came on pretty much as the chemist of sorts making these. And I had a 10-year career where that's what I did. Somebody would bring me an antibody and I'd put it on, pretty much doing the same thing over and over again. This worked out very well for the immunologists. In days, I could make things that would keep them happy for months because the animal experiments took forever. So they got things they wanted, and I could disappear as necessary to do whatever things I wanted. So I really got it as a pretty good racket from a publication point of view. I was middle author on dozens and dozens of things. Somebody would bring me an antibody, we'd do this, put them in different combinations and stuff. It was pretty much the same thing over and over again. In thinking about the longer range of my career and the things that I've done, what this really focused on is what's called multivalency. That is the ability of when you have a lot of the same thing, let's say the surface of a virus, when it comes in and interacts with the B cell, it's doing the same thing. It's bringing in a lot of these receptors, cross-linking them, activating things. And so one of the ways this works for these polymers is when one end of the polymer binds, well, now you've reduced the problem from 3 dimensions, where one antibody is flying around trying to find something, to 2 dimensions because you've tethered one end of this. And so the effective binding constant goes up by many orders of magnitude. And in turn, the ability to signal across that membrane goes up by orders of magnitude. It's a reduction of entropy in that you've reduced the dimensionality. The problem with my working on this for so long is that, as I vaguely recall from PChem and thermodynamics, is that entropy needs to be constantly increasing. And so if in one part of my life there was some reduction, however small, it had to increase in another part of my life. This is where my wife's high degree of organization and focus comes in handy. Now, what I was doing was putting antibodies on polymers forever. And some of the controls were we would put random proteins on, things of no real value. I should say the polymer had no real value. It was just a scaffold. So there I was putting valueless things together. What I learned was, so essentially I was putting chemically linking proteins to these carbohydrate polymers. And what I discovered was I was speaking prose. What I was doing was actually synthesizing what are called conjugate vaccines. These are vaccines that are where you take a protein that may have value and a polysaccharide that has value, chemically link them together, and you make this conjugate vaccine. So generally, the polysaccharide part is part of the outer fuzzy membrane of certain bacteria. So these are composed of carbohydrate polymers, repeat units of particular sugars, and every bacteria has its own arrangement of the sugars. Some of the diseases that these are involved in are strep pneumonia, Haemophilus influenza B, and I didn't try to correct you there on your pronunciation, Salmonella typhi, and Neisseria meningitis. One of the reasons that I could not really think about going into microbiology is I can't pronounce most of these either. Another reason I can't go into immunology is I just can't remember one fact from the other. Nice thing about my life now is I work with immunologists who they can remember which cytokine is which and which thing goes up and down, but I don't have to. Now, a problem with carbohydrates, if you can make antibodies against these capsular polysaccharides, you can protect against the disease. The problem is that these polysaccharides are very poorly immunogenic in infants, unless, well, so these diseases are devastating. Now, I had some question with myself about whether I should show this next slide. This is the land of anti-vaxxers. Even among highly educated people, there are still anti-vaxxers. These diseases are not ones that you want to fool around with. Even simple childhood ones, measles, everybody my age has had measles. These diseases are devastating. They can be prevented with vaccines. This is an example of what happened with Haemophilus influenza, HIV vaccine, that every kid now should get. Okay. You can see the reduction in disease. Now these, and this is true for all of these vaccines. The three that are, the big two that are there now are Haemophilus influenza B and the strep pneumonia, Prevnar, if you've seen the ads for that. The Neisseria, because of the relatively low prevalence among young kids, is not as big an issue. It is among college kids, and you've probably heard of that. We heard Princeton, and that's really why Princeton's most in the news these days from their meningitis type B outbreak. But these so-called conjugate vaccines are among the most complex and most expensive of all the childhood vaccines. The strep pneumonia vaccine, for example, costs about $100 a dose in the US, 3 doses. It's about the income of some countries. So I learned I was making these. The group said, well, I'm the chemist, so this is what I was supposed to do. Going back to those qualities that I had burnished at Harvey Mudd of anxiety and security, as well as new ones I picked up, fear of doing new things and unknown and so forth. Part of that, the chemistry that was used was not much published, and it was very dangerous chemistry. So I used another piece of advice that I learned at Harvey Mudd from Ron Vaughn. These days, it's 5 minutes on the internet is worth a month in the lab. But pretty much, I've never worked, for most of my career, I've never worked in the same place as my supervisors. So all the immunologists were a mile away from where I was. So really nobody knew where I was. So between just simply not being anywhere and also the fact that it's cooler in the library, I spent a lot of time just wandering around there. And I found a method in another field that I thought could possibly be applied to making conjugate vaccines, to activating the polysaccharide, linking the protein. This little molecule here changed my life. Suddenly I went from, pretty much suddenly went from somebody who was kind of drifting in life, doing okay, but not really having any purpose, to really having something that I wanted to do more than anything else. At the time, my wife was on bed rest, 7 months of bed rest. I could only work at night. By the way, if you want to know what 7 months of bed rest is like, think of a flight on Southwest for 7 months. And having me as the steward. So I could only work at night. So I'd order these chemicals, and for reasons that were, I mean, nobody in the vaccine field would ever try this, but because of the other stuff I was doing with worthless materials, I happened to have them under conditions that really couldn't fail. And I mix this stuff together, it makes Jello. This was like 10, 11 o'clock at night. I'm the only one in the university, probably, and make Jello, and that was the eureka moment. It worked. Pretty much the rest of the year was backtracking from that, trying to make something that was soluble, that would work, and that would be usable as a vaccine. But seeing that it worked, that moment, and other people had actually tried this, but I failed I succeeded because of my complete ignorance. I developed this term of creative ignorance, really too stupid to know any better, so I tried it. And it really made a difference. Very quickly, this technology got licensed through a set of really random, completely serendipitous events to GlaxoSmithKline. One of the requirements of completing this was that I needed to demonstrate that the method worked for an actual real vaccine. Fortunately, it worked for them the first time they tried it. It took me close to another 20 years before I succeeded with that particular thing. That was part of the requirement for them to license it. So that was a little piece of dumb luck on my part as well. Well, this application, finding something focused, also gave me a little financial security as well, thanks to the change where the universities were allowed to share with inventors and own, that they could own an invention that was funded by the federal government. So that really set off, gave me some financial security to start off in other directions. And I gradually began to transition to private, some small biotech companies, still working on the same kind of stuff. Through a large part of that period, I had very little supervision on my patents and my applications. So this was really the most creative part because I had one piece of successful track record. They would just, whatever I thought of, okay, let's go file a patent on it. So I had lots of great ideas, but most of them are really not suitable for anything. They look great on the wall, though. And then I was still part of the university, so I had that, and then I transitioned to another company. The first time in my life where I was part of with my supervisor in the same facility where I was, and things kind of went downhill there very quickly. And another person that I'm extremely grateful to, my supervisor from the university, who was the scientific director, that despite how much trouble and headache I caused him, and there was a significant amount of it, that he didn't fire me and kept me on. Some of the lessons I learned there was I really make a lousy employee. I do make a great collaborator. I've always found it's much more fun to help people with their work than it is to do what you're supposed to do. And also part of this, I was in a position where I could recognize that I needed to start taking responsibility for myself if I was ever going to actually grow up. And it's not really something that was news, that if this is what I needed to do. We're going to come back to Julie on that same point. I think it should be fairly obvious. But there's something intrinsic. It has to come from within you in order to actually make that change. So as part of that, now I should just say one of the reasons, many, many reasons why I made a lousy employee, is that if I ever thought of a joke, I had to tell it to everybody. So when I thought of something like this, at this event, I had to do it. This is the famous color-changing rabbit. Yeah. Now actually, that's the easy part. See, dyeing your hair is easy. Changing the sex of the rabbit, that was the hard part. This is Miss Molly Mischief, my current rabbit. She's world's second greatest hairpiece. So 2006, I had another tip-off from somebody that if I didn't quit, I'd get severance. And in summer of 2006, I was laid off along with most of the rest of the research staff and told to stop being so happy. Because it solved the problem of when should I quit, because I didn't know, how do you explain to your wife you're giving up all these benefits for nothing? The main focus of this, and over the years, through the conjugation chemistry, the fact that I had licensed, the stuff, people knew that, Glaxo was using my stuff. The other work that I had done, I had made friends with the director of the largest vaccine company in India. And so we had talked about it. And shortly after I started my company, he gave me first contract and agreement that let me start building the company. Now we do have, this is kind of, if I'm being serious, this is what I'd say, but our actual mission statement is a little more focused. And it's really critical. I wanted to do good. Glaxo and Pfizer, the manufacturer of the strep pneumonia vaccine with their $100 dose per dose, I knew the people who made that decision. It had nothing to do with markets or what's the value of this, it's what they could get away with. And that really pissed me off. That these are not potato chips, these are not apps, it's not whatever you can get for them. These are things that save lives. I was in a position to do some good. I had enough, I basically had a salary, I didn't need that part to start the company, I needed to run the company. And this is what I wanted to do. I felt like I was in a position to do it, and I really felt a moral sense that this is what I should do. So doing good, having fun, I really needed to have fun, that was and if anyone's tried to run a company, you know that you need money to run a company. So that was the third part is a critical part and really one of the most difficult parts. Because in biotech, it takes a long time to bring a product to market. The first vaccine I worked on took 13 years to get to market. I know because I went from the hospital where Julie was delivering Elizabeth to the patent office, and it was licensed shortly after that. And I know, so that's kind of tracking it to my daughter's birthday makes it easy to keep track of how long it took to get to market. Another part of this was, as a self-funded company, I didn't want to take directions from anybody else. And so I call it a not-for-much profit company. This concept is now out there. There are these, what are called type B companies, like type C corporations, B corporations, are companies that have public good as their major aim. I didn't know about any of that, that's just what I had in mind. By being a not-for-much profit company, I didn't need to return millions just to an investor. I just needed to make a living. It's much easier to do that than to try to get rich. And so that was really the focus. As a not-for-much profit company, I could go to other companies in India and in China and talk to them, and it quickly became obvious from my attitude that I was not out to extract as much money out of them as they could. The work I did with them was as a goal, what's the least I could do this work for, not what was the most I could extract out. And that's been very successful. And that process from the first contract has allowed me to slowly build my company. Let's say we now build it organically. We did try to build it inorganically, but we found they just threw the rocks back at us. So I have a terrific crew of people who work for me. I've got now somewhere 6 to 8 employees, depending at the time, and they're just terrific. And they're really the ones who have helped me grow the company. I've taken the lessons about how I want to grow, what kind of a mentor, people who have mentored me, and I try to be the same to others. I try to run the company as a cooperative one. Don't look at it as try to help other companies. I'm the go-to guy for a lot of different, for resources and other things. And I found that process slowly has helped me to build the company by not asking for things. People come back and help me in many ways. And I'm very appreciative to the people who actually now do the work in my lab. Since fundraising is an important part of this weekend, let me say that when I give money to the university, to the college, and particularly to the chemistry department, I always make it for general funds. I remember being told everybody gives money to get their name on the door, on the instrument. I like to give my money to help replace some of that broken glassware I left behind. So the Serum Institute of India is one of the companies I've worked with. These are now moving into clinical trials. We anticipate that there'll probably be on the market sometime around 2019, 18, 19, depending on approval. The Chinese, the Chengdu Institute, probably in the early 20s. Out of this, I now work with many other companies. In terms of this field of bioconjugation, I'm a go-to person because I'm easy to work with. Just as an example of this, with USAMRIID and the anthrax vaccine, they had heard about me, they came, we talked about it, and said, well, that doesn't look like a very hard problem. And go, well, you know, if you want to do a DoD contract, there's going to be a lot of work. You know, it's just, you know, I said, well, I'd rather chew my extremities off than do a DoD contract. So I'll tell you what, we'll just do it. So they have a $3,000 limit on their credit cards, and we sent them, nobody here from DoD contract, and we sold them an awful lot of little vials of water. But we did it on our first try for the first $3,000. So now it becomes their problem to figure out how to get the money to us, to actually go forward and make the contract, to make the vaccine. And that's kind of the way a lot is, by being really easy and enjoying the work, making it fun, been able to generate a lot of other interactions. Being on the university part, I don't have to know any biology or any of the other stuff. They do that part. They're really good at writing grants. They've got all of the experts, and so I just come in as the third wheel. So just as my academic days, publishing papers as a middle author, I got a nice little niche where somebody else does a lot of the work, and I do my little bit that I do best. Coming to the last part of our mission statement, trying not to go broke. As we've grown, going from a little lab bench about this big to now about 5,000 square feet, and just a major operation of trying to keep it going. You know, initially I used what's called the toilet bowl method of accounting. As long as your account looked flush, you were good. It's really got, a lot of it's gotten beyond me, so you know you need to start hiring people to manage this and that. So trying not to go broke has become a major part of it. Now, I have made fun of apps and some of this other stuff and you're making money, but I'll say Google Words is where this came from. Following my usual habit of doing nothing, so I'm just Googling the polymer that these antibodies were put onto. Came up with this, oh, there's a company I never heard of. I send them an email, I get a call back, we talk for about an hour, strike up a friendship. So I'm now in this new field of industrial biopolymers. Turns out these same Dextran polymers that we're using for everything are used in aluminum smelting, fracking fluids, wastewater. They serve as flocculants, friction reducers, proppants in the fracking fluids and stuff like that. So we're now in the area of environmentally friendly fracking fluids. So these are really cool. If I ever have to write and I'd like to write an article on you know, the unknown value of government, federally funded research. I've gone from studying the immune properties of B cells to this. So it's the sex life of B cells. So these things they're really cool. They're really weird. A lot of this pond scum and stuff that make this stuff. And so we have the microbiologists. We have from the vaccine stuff characterizing these big polymers. We have some of the rheology stuff, light scattering stuff, other things that come back to that biophysics degree that I got that I can't remember much about. But you can hire people who do. And so we're into this area. So we're now moving towards this plant. This guy's got 150,000, multiple 150,000 liter fermenters, and we hope to be having products there by the end of the year. Would say vaccines, carefully, highly regulated, fracking fluid, not subject to environmental water regulations. They don't care. We make something, down the hole it goes. Hopefully these environmentally friendly ones will make the world a little bit safer. I'm also hoping to get into the wastewater treatment area as money starts coming in to fund some of these other random areas. So for the future, we intend to keep focusing on our mission statement and having fun.